Hello whizzes. Welcome to my channel. Today's topic is humoral immunity. Humoral immunity is a crucial component of the adaptive immune system, responsible for defending the body against pathogens, primarily bacteria and viruses. This immune response involves the production and actions of antibodies, which are also known as immunoglobulins, Ig. To understand humoral immunity in detail, let's break it down into several key aspects. B cells and antibody production. Humoral immunity primarily revolves around a type of white blood cell called B lymphocytes, B cells. B cells are produced in the bone marrow and have specialized receptors on their cell surfaces that can recognize specific antigens, which are usually proteins found on the surface of pathogens. Antigen recognition. When a B cell encounters an antigen that matches its specific receptor, it binds to the antigen. This binding triggers the activation of the B cell, leading to its proliferation and differentiation into two main types of cells, plasma cells and memory cells. Plasma cells. Plasma cells are responsible for antibody production. They are highly specialized and produce large quantities of antibodies that are specific to the antigen. Antibodies are proteins that can bind to the antigen and neutralize or mark it for destruction by other immune cells. Antibody structure. Antibodies are Y-shaped molecules with two identical antigen binding sites at the tips of each arm. The constant region of the antibody, the stem of the Y, determines its class, IgM, IgG, IgA, IgD, or IgA, and various effector functions. Neutralization. Antibodies can neutralize pathogens by binding to them and blocking their ability to infect host cells. Opsonization. Antibodies can tag pathogens for destruction by phagocytes, e.g., macrophages, through a process called opsonization. Complement activation. Some antibodies can activate the complement system, a group of proteins that enhance the immune response by promoting inflammation, cell lysis, and phagocytosis. Antibody-dependent cellular cytotoxicity, ADCC. Antibodies can facilitate the destruction of infected cells by natural killer, NK, cells. Memory cells. Memory B cells are long-lived cells that remember the antigen they encountered. They remain in the body after an infection is resolved, providing long-term immunity. If the same antigen re-enters the body in the future, memory B cells can quickly generate a rapid and specific immune response. Isotype switching. B cells can switch the class of antibodies they produce, e.g., from IgM to IgG or IgA, through a process called isotype switching. This helps tailor the immune response to the specific pathogen. Primary and secondary immune responses. The first encounter with an antigen, e.g., during an initial infection, leads to a primary immune response. Subsequent encounters result in a secondary immune response, which is more rapid and effective due to the presence of memory B cells. Vaccination and immunization. Vaccination is based on the principle of humoral immunity. By introducing a harmless form of an antigen, e.g., a weakened or inactivated virus, the immune system generates memory B cells that can provide protection against future infections with the actual pathogen. In summary, humoral immunity is an essential part of the immune system that involves the production of antibodies by B cells in response to specific antigens. These antibodies play a crucial role in defending the body against infections and providing long-term immunity through the generation of memory B cells. Understanding humoral immunity is fundamental for medical students in diagnosing and treating various diseases and designing effective vaccination strategies. If you like this video, please like, share and subscribe to my channel. Thank you for watching.